Hi, my name is Monica Ponder. I'm an associate professor at Virginia Tech in the Department of Food Science and Technology today. I'm going to talk to you about some of our recent work investigating how the inoculation method of Salmonella enterica affects the survival when it's placed in low water activity environments. Specifically today we'll be talking to you about peppercorns and whole black peppercorns that have been stored at a water activity of 0.3. In this experiment we had 4 different methods. In the first step we had a planktonic state method, so the cells were grown in a TSB medium, so that's a nutrient-rich medium, and then they were just spotted onto the peppercorns, and then we dried that back down to the water activity that you would expect to find of a spice within the commercial environment. In our second method, we took cells that we had grown on a petri dish. In the microbial world, we would refer to these cells as sessile cells, and then we scraped them off. The nice thing about this method is it gave us a lot of cells, and so it gave us a lot of starting inoculum to work with. And when you're investigating something like a challenge trial, that's really important to have enough organisms to start with in the beginning. Our third method of comparison was a dry inoculation method. And really with that, what we're talking about is the ability to transfer Salmonella from a carrier molecule that we did the inoculation of it. In this case, we inoculated a silica, a sand particle, and then we mixed that in with the peppercorns. And so the contaminated sand molecule is actually what was transferring that Salmonella onto our peppercorns in that phase. Our fourth inoculation method is the ability to develop a biofilm of Salmonella around a peppercorn spice kernel. And we did that by incubating the Salmonella in a nutrient mixture along with the peppercorns. And we found that after time, and really only 24 hours, there became large numbers of microorganisms that were on there. And they weren't only just Salmonella. So Salmonella was becoming part of this large consortium of microorganisms that survived around that spice particle. And so in order to get any non-adherent cells, so that weren't part of the biofilms, we washed them several times. And so we would take some nutrient-free media, phosphate buffered saline in this case, and add the peppercorns to it, and then just shake. And that would remove any of the cells that weren't adherent to that. We would then dry down all of the peppercorns after that to that starting water activity that we see normally in commercially sourced peppercorns. So after this, we then determined what were our number of surviving Salmonella on a selective media. And we used XLT4, which is a selective media that reduces the growth, inhibits the growth of other spice-associated microorganisms, because spices can have fairly large microbial loads that are associated with them, in addition to the Salmonella that we know we just inoculated them with. And so by using this XLT4 media, we know that it represents only the Salmonella that are surviving on those plates. And we compared what happened after 28 days of storage within a desiccated environment. What we find are the blue represents the biofilms, the red are the dry transfer, the green are the wet broth, so these are planktonic cells, and the purple are the wet scrapes, so the sessile cells. And our plot shows the log reduction from the initial counts. And as you can see, there's quite a bit of difference, particularly with these green bars here. So these are our planktonic cells that have just been taken from a nice, happy state, growing within tryptic soy broth, and spotted onto the peppercorns and then dried. And so you can see the largest reductions in numbers of Salmonella that were recovered on the XLT4 media are with this, while several of the other inoculation periods had very small reductions. So we hope that the research community, and that includes academics, that includes people in the regulatory industries, and that includes people in companies who are all interested in validating their low water activity types of processes and inactivation strategies. I'm hoping that they'll take from this work that they really need to be cognizant, and we need to be developing standardized methods that everyone is using. And those standardized methods are also considering the origin of the strains that are being used. So the next place that we will go with this is working on developing methods for inactivation of Salmonella when they're in their hardiest state, and using ethylene oxide and using steam treatments. After we have developed identified conditions that have a nice reduction in Salmonella, we want to determine that the spices are still of quality that industry requires. And so we have a sensory component where we have a sensory scientist who will be comparing with non-treated spices versus the treated spices to see if a test panel can identify differences within them.